All right, everybody, we're going to go ahead and get into biotechnology. So let's go ahead and take a look at this. All right. Uh, Basically, in short, when we're looking at biotechnology, we're looking at how we can go take foreign DNA in order to put into another organism to be able to start to transcribe and translate a gene it didn't originally have. There's lots of different reasons or uses for biotechnology. So this is going to be a general understanding of it, how it ends up working, and our little processes of it. And I'll try to explain it using the biotechnology lab we conducted in class today. So, in short, biotechnology is just the process of manipulating genetic information within an organism. All right, you're basically sorry. So you have like a bacteria right here. So you got bacteria. Okay, so we got bacteria right there, and we have, I don't know, let's just say like a plant, right? And you got a plant. All right, and in each one of these guys, there's going to be DNA associated with it. Geez, of course I end up separating out. Let's go ahead and draw that again. So bacteria, and we'll go ahead and do this little squiggly guy, right? And then we'll go ahead and get our plant. We'll actually do the plant green this time, so it actually works a little bit better. Right, so you got your plant, right? And what you're more which just trying to do is a process in which I'm going to go and isolate some section of it, maybe like this section of the DNA. And what I'm going to go and do is basically isolate it, cut it in order to eventually maneuver its way and engineer it so that way it's then part of the um, bacterial genome. And once it's in the bacteria genome, whenever this bacteria is going to go and transcribe, the DNA into mRNA and then translate it into a protein, it's now going to be creating this this gene right here, this gene that we were taking a look at that it wasn't able to go and do. So in the case here, maybe we just gave it the ability to become photosynthetic. Um, that it could be one specifically. Maybe I can make it start to create a protein it wouldn't normally be able to go and do. This is how we end up creating all the insulin necessary in order to go give people who are diabetic, right? Like we force the organism to go and make the stuff synthetically. There's lots of little other crazy things that are out there. Other examples of stuff, and you can go look this up online. There's a luminescent animals like cats, where they basically ended up putting making glow in the dark cats. You can go take a look. I believe this was in Japan that they ended up doing it because they, I guess they had nothing better to go do. You have goat milk with spider silk. Right, so now we end up having a spider silk protein being able to be made within goat milk, so that way you're able to mass produce it. It's a lot easier for to make products. Other things that I can do: pesticide resistant crops. I can end up making it being that I can go spray them with this really nasty chemical that would normally kill the plants, but then I make them resistant to it. And what happens, that ends up killing all the pests, all the insects that are on it, right? So that's all biotechnology is. I'm just trying to manipulate the genome of an organism to make it do something it wasn't able to go and do before. So in the case here in our class, right, we ended up, I'm going to do like a reset, right? We ended up giving you E. coli, and we are adding what is called the um, pGlow gene or the vid gene. And what's supposed to happen is that when we go and take a look at our petri dish, and we go and take a look at our E. coli that should be up on here, hopefully this ends up working correctly. It's going to go and start to glow in the dark once I end up, throwing UV lights on it, right? The ones that end up getting the gene will end up glowing in the dark. So really, really cool stuff. So that's what we're hoping to accomplish next time we see them in class. So the first thing we end up doing is that we're going to go ahead and use gel electrophoresis in order to separate out the DNA to figuring out, our sequences to begin with. Now, for those that are not aware on how gel electrophoresis works, it starts off like this. We end up putting the DNA into this side over to here, and you attach a negative cathode onto its end and a positive anode on that end. And what's going to happen is that the DNA we can see in this picture right here is it's going to be moving in this direction from this initial location over here. And now what ends up happening, the reason why it's going over there is that it's attracted to this positive anode over there. If you go and take a look, right, our nucleic acids, right, with uh, our ribose or deoxyribose sugars, right? So in case of DNA, it's deoxyribose. Are nitrogen bases, but really what we want to look at is this phosphate group. Because what do you remember about phosphate groups, right? What do you remember about phosphates and charges? Think back to like ATP, right? What are all these phosphate groups supposed to be? Hopefully you remember that they're all negatively charged, really, really super negatively charged. So basically what happens is that because they're negatively charged, this negative charge part of the DNA is interested in moving towards the positive end on that side. So the DNA is going to go travel to the positive end. It's kind of entertaining that when you get students who end up setting this up and they botch it because they add the anode over on this side and then the cathode over on this side, and the DNA goes in that direction. Like, what did I do wrong? And it's like you set it up incorrectly. So your DNA literally traveled backwards and basically fell off on the other edge of the gel over to here. So yeah, always make sure that they go over there because of the phosphate groups. So that's why. Now, one thing that people aren't aware of is these separation bands, right? So, or at least why these separation bands work. So what happens is that the DNA is going to be added with these things called restriction enzymes, and it's going to go and explain what these restriction enzymes are. Is that let's say that we had TAGGC ATTG Mm-hmm. Just doing this right now, just so that way I can kind of give you an idea. All right, so this is my DNA sequence right here. Okay, what a restriction enzyme looks like is that it's going to be basically a protein that's going to go and have a complementary base pair on it. So this right here is going to be the restriction enzyme, and as you can tell, right, our T's will pair with A's and the G's will pair with C's. I guess actually in the case here, you can also use these for mRNA as well. So your T will pair with the A's, and then the A's will pair with the U's in the case in here. But how this restriction enzyme works is that it looks for a location where it's complementary, right? Where we see right here. And what's going to happen is that it actually breaks this sequence of DNA at that specific point. So it restricts the size of the sequence, and it does it for every single time that it sees that. So when I go and take a look at this again, I'll see that this sequence specifically shows back up at this point right here, and this sequence is very, 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 very particular. So it's only going to go and break those ones, right? So this could be anywhere between like maybe like 4 nucleotides to like hundreds of nucleotides in between this section and this section that's being broken up by the restriction enzyme. All right, so it's going to go cut the DNA into many, many, many different sizes. You'll have long pieces, short pieces, medium pieces, more long pieces, even longer pieces, and maybe like a bunch of little tiny shorter pieces, right? It doesn't really matter, just that the restriction enzyme ends up breaking it. That's what we're saying here. The DNA molecule in different sizes is due to these restriction enzymes. And now what happens is that all this cut up DNA is going to make its way into the wells, and that's sample one. Another sample is going to go into sample well two, and another sample is going to go into sample well three. And what you're going to go and see is this thing called a banding pattern over here. The banding pattern is how the DNA separates, and what happens is that the longer molecules, right? These, let me actually change the color so we can actually, you can see the ones I'm talking about here, right? The longer molecules are like these guys over here, like this one and this one. They can't travel so far because they're so long and they're trying to move through this gelatin. Like imagine that there is a crowd of folks, right? And you're like at like a party or you're at prom or whatever, you just there's people everywhere, right? Big old crowd. Okay. And let's just say hypothetically, it's like you and your friend, right? So here's you with your friend right next to him, right? You're holding your hands and you got to make your way through the crowd, right? Well, you and your friend are only two people and you have to hold hands the whole entire time. It's relatively easy for you and your friend to travel quickly to the other side in order to go and get there, right? Now, as opposed to maybe a bigger group of friends where maybe it's like, now we end up having like, the shoot, let's just go and say 10 folks, right? So just use your imagination and then we can get like 10 individuals right here, right? So a lot of folks, right? They all have to hold hand in hand and hand in hand in hand. And now they have to go, well, it's going to go and take way longer for all of them to be able to move and meander forward because there's so much more that they have to go and try to, move around in order to actually, get to the other side. So the longer ones, right? The longer ones basically stay over here on these regions over to here, while your shorter patterns, right, the shorter patterns like we had like right here, they're able to go out a lot further. They it's a lot easier for them to move through the gel as opposed to the longer ones that are over there, and that's what we end up seeing. Now why we're talking about this is that let's say that vid gene that we're talking about, the bioluminescence gene, well, I know when I cut it, and now I've cut the DNA, well, I'm going to go and test all of these different sequences, and I'm going to go find out, here's where the vid gene actually is. So I know that vid needs to be this specific banding pattern right there. So now I've isolated my gene sequence and to be able to go ahead and start to put into my organisms at that point. All right, let's go ahead and move on to the next one. All right. So in order for me, now I ended up getting my genetic sequence. Now what I need to do is copy it. I got to mass produce it. And the way that this works is by doing a thing called PCR, polymerase chain reaction, or PCR for short. It's basically a process that it's DNA replication in a test tube. Like if you wanted to, right? Because keep in mind, right? Normally DNA replication happens during the S phase. Well, what you're basically doing is that you are creating conditions in which you are going to go and just copy the DNA over and 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 over again to the point that you have so many sequences. And this is the process, right? You denature, right? When you separate it, you anneal. Then we end up adding primers onto it. Then we extend them into new sequences, and then we basically do the thing all over again. And then you denature and anneal and extension, then you do it all over again. And you're probably wondering, well, why? How does this make a difference? Well, watch this, right? Okay, so you start off with one sequence of DNA. All right, now we're going to go ahead and PCR it. Now we doubled it. Okay, cool. Now we're going to go do it again. We double it again. All right, now we double it again. Now we double it again, and we double it again, and we double it again, and we end up doubling it again, right? And you could probably end up seeing what's going to happen here, right? We're basically getting to the point that we exponentially grow the amount of DNA that we can work with, which is why when we looked at the little plasmid guy, right? When you saw like the little plasmid from the experiment, this was just nothing but that DNA sequence, right? That was that DNA sequence located within this thing, and then you use your inoculating loop in order to go and grab it, right? So we just basically copied it over and over and over and over and over again. Why? Because one, I want to give you more of it than you really need, because what we're hoping to do is make sure that the new gene takes. We're trying to make sure that you actually get the information so that way it's going to be a successful transfer. That's why I was saying that maybe some of the groups will have glow in the dark bacteria while others may not. So we use PCR in order to just make sure that we can make sure we have all the stuff. This is just all the different things that go into it and how it works. So basically your DNA, your primers are there in order to go ahead and start the sequence. Your nucleotides are there to go build the new DNA. This polymerase is going to go ahead and be used in order to go ahead and make the actual new sequences of DNA. The buffer is just there for a solution to work with. And guess what? You do it in a PCR tube. Like all this stuff together, and this really cool experiment makes this whole process happen. So neat. Okay, so then you have bacterial transformation. What the heck does bacterial transformation is? This is literally just DNA going into a bacterial cell. All right, the idea behind it is that bacteria will go take any DNA that it's able to, and then if it's able to input it as part of its genome, it's going to go ahead and do it. So this is what happened here, right? Here is that DNA that we had within the little test tube. We put it into the plus plasmid, right? The plus plasmid sequence, and we heat shocked it. Well, why are we heat shocking it, right? Then I tell you, it's like your pores. So we're trying to get it so that the plasmid will go into one of these cells, hopefully, and then if it goes into the cell, cool. Now we can go ahead and put this genome into or we can put the plasmid into the bacteria genome. Bacteria are the only ones that are able to go and do this, for which you need to be aware of, and it's great, cool. They're able to now go and make that protein, and now hooray, we are now able to actually go and create the stuff. Now, bacterial transformation just doesn't do the glow in the dark gene, right? There's so many more applications to this. For example, you can end up having bacteria becoming superbugs. Where they could potentially end up getting an immunity to an antibiotic that they didn't have before. So we're actually also going to be making our bacteria ampicillin resistant. Which is not really that good because ampicillin's a good antibiotic, and we're actually forcing a mutation that's going to go, or a mutation, I should say, we're forcing genetic engineering it in order to become more virulent. So that way, there's one less thing that we can use in order to go ahead and deal with it at that point, right? Other things that can end up happening is that it can go and create other proteins or other yucky, nasty stuff that could potentially cause more harm to hosts and stuff. So, bacteria transformation, like as long as the gene exists and they take it in and they're able to incorporate into their genome, if it gives them an edge, they're going to just be doing it, and it's just wild to think about. And this right here is just showing you DNA sequencing. So in ye old days, they would end up reading the banding patterns to see the actual DNA sequences. This is how the Human Genome Project worked. We are beyond this at this point. We don't really look at this anymore. They now have these devices that end up shocking the DNA as it's going through this really, really small pore, and they shock it and they measure the electrical impulse, and that tells you if it's an adenine or a thymine or a guanine or a cytosine and stuff. It's really cool things, but this was just basically our way of being able to figure out the sequences. And again, once you know the sequence, and you're able to go figure out how this whole thing's going to go and fold. You go and translate it. You transcribe it into your mRNA, and then that mRNA would be translated into your protein. You can come up with an idea in these models on what you're going to be able to go and make once you end up throwing these nucleotides into their respected positions. So lots of really really cool stuff. All right, that's all I got for y'all. We'll do the tests and everything else, and catch you on the next one.